Okay, so good morning everybody, and thanks a lot for taking the long trip to join us. It's really a pleasure to host you here. So I'm going to tell you a little bit more about dendritic cells and their key role in orchestrating the immune response. They've been mentioned in a previous presentation briefly, but I'm going to tell you all the secrets about this amazing cell type. So dendritic cells arise from a common myeloid progenitors like macrophages, but unlike macrophages, which are mostly dedicated to destroy pathogens upon internalization, dendritic cells provides a critical link between innate and adaptive immunity, and their main function is to activate the T cell and B cell response. These cells were discovered in the early 80s by Ralph Steinman, who got the Nobel Prize for his findings. And actually, the scientists at the time were looking for the accessory cell that would explain the observation that if you take an antigen and you just mix it to T cells, you get no activation. But if you add cell suspension from a mouse spleen, you get a response. So by fractionating down and dissecting what was that component, they end up in discovering dendritic cells. And they call them dendritic cells because of these dendrites that they have under a microscope. So dendritic cells are so good to activate T cells because they have a very high endocytic capacity and express a vast array of endocytic receptors. They have a specialized antigen processing machinery, which I will go back in a few slides. They can migrate out of peripheral tissue to reach lymph nodes, and once they are in this mature stage, they express a lot of co-stimulatory molecules and secrete soluble signals. And these three signals altogether, MHC peptide complexes, co-stimulatory molecules, and soluble signals, are very critical in activating, as I said before, T cells, B cells, and NK cells. However, they are not a simple cell types. People have been discovering, they keep on discovering subsets of dendritic cells, which are quite complex. They are located into different, they have specific locations and quite diverse functions. But just to sum up and make it simple, I'd like to remember that in the skin, in peripheral tissues, we basically find Langerhans cells in the epidermis, and we find CD11b and CD103 dendritic cells in the dermis, which may be derived by a pre-DC precursors, or CD11b DCs that derived from monocytes during inflammation. If we now move to skin-draining lymph node, here we'd find two major populations, the ones that are residing in the lymph nodes, that are CD8 alpha positive and CD4 alpha positive, and those that migrate, that are recently emigrated from the tissue into the lymph node. So again, we find CD11b and CD103, and I will tell you something about these cells again later on. As I mentioned, one of the most important specific feature of dendritic cells is their antigen processing and presenting machinery. So if you think at the T cell response, normally antigens which are internalized from the environment are presented on MHC class II molecules to activate CD4 positive T cells. However, dendritic cells also have evolved a mechanism to present peptides derived from exogenous antigens onto MHC class I. This phenomenon is called cross-presentation. It explains the fact that these cells are absolutely required in vivo to activate cytotoxic responses against virally infected cells or dying tumor cells. People have studied a lot in the last 10, 15 years how this mechanism of cross-presentations work, and I'm just summing up some of the most important features of this specialized antigen processing machinery. One very interesting finding is the fact that the phagosome in dendritic cells is kept highly alkaline. So if you think a macrophage has a pH in the phagosome that is about 5.5, so it's really devoted to activate catepsins and proteases and destroy the pathogen. Instead, dendritic cells have developed a way to keep the pH quite high. So antigen degradation is slowed down and epitopes are kept intact for cross-presentation. So somehow they slow down the degradation to allow more antigens to be presented. And this is, of course, ensured by a complex trafficking of organelles that maintain the proton pumps active in order to keep this pH at the right level. So once antigens are processed in the phagosome, they can be either shuttled out in the cytosol past the proteasome and be loaded in the ER to be sent on the surface, or directly loaded in the phagosome to be presented on MHC class II molecules on the surface. If we want to put all I said in a context, in lymphoid and non-lymphoid tissue, this cartoon explains a little bit what's going on in the body. So dendritic cells are in the steady state mostly located in peripheral tissues, where they patrol the environment for the presence of pathogen. Once they have captured a pathogen, they migrate in a CCR7 dependent mechanism to lymph nodes, where they can present antigens to CD4 T cells, cross-present antigens to CD8 T cells, or induce B cells to undergo class switch recombination. Another important concept about dendritic cell biology is the fact that they are not only involved in activating responses, but they are also very important in maintaining peripheral tolerance. And this is especially well documented in the gut, where diseases are really important to ensure gut homeostasis and tolerance against food antigens and commensal bacteria. And this has been elegantly shown by experiments where you deplete dendritic cells in the gut and you have no tolerance against food antigens anymore. So I told you about the fundamental biology of dendritic cells and their ability to really orchestrate different aspects of the immune response. Explains why they've been largely employed in medicine. And one of the most probably relevant settings is cancer immunotherapy, which is simply summarized in this cartoon, but not only, as has been mentioned before, they can be used for vaccination against viruses. So here are listed 4 major situations in which dendritic cells can be used to mount efficient immune response against tumor cells. One possible passive mechanism is the fact that during chemotherapy and radiotherapy, the so-called immunogenic cell death provides antigen that are captured by dendritic cells that then can initiate a response. A second active way of exploiting dendritic cells for therapy is the possibility to isolate them from peripheral blood, load them with tumor-derived or viral antigens and infuse them back into patients. A further development upon this method is the possibility to target directly dendritic cells in vivo by using antibodies that are targeted to endocytic receptors expressed on dendritic cells fused with tumor antigens. Or still, this is probably the newest aspect of this research, is trying to reprogram the microenvironment and dendritic cells towards cancer destructions instead of tolerization. So in my lab, we are studying dendritic cells in what I like to summarize in three different situations. The first one is trying to understand the really basic cell biology of T-cell priming. And the second and the third are to study dendritic cells functions in 2 pathological contexts. One is a primary immunodeficiency, and the third is what happens to dendritic cells during cancer immunosuppression. So when dendritic cells reach the lymph node, the first thing they have to do is to find the right T cells and establish a tight interaction, which is called the immune synapse. This is something very dynamic, and I'm going to show you a movie of how DCs, it's an old movie, how DC are really jumping and undergo a lot of remodeling of their membranes to capture the T cell. Well, the big cell is the dendritic cell, the little one is the T cell. And we've been able to prove many years ago that indeed cytoskeletal remodeling in dendritic cells is essential to this function, as indeed if we knock down an important regulator of actin polymerization, the DC, it's really paralyzed, and T cell activation is really abolished. So once the interaction is formed, how are pathogen-derived signals transmitted to T cells? We have to think, as I mentioned, there are basically 3 types of signals: MHC peptide complexes, which gives an idea of the specificity of the antigen, and co-stimulatory molecules or cytokines that instead give to the T cell all it's needed to keep on proliferating and differentiated into an effector cell. So we've been interested, as I mentioned, to the cell biology of this phenomenon, and we've been able to show some years ago that dendritic cells, when they interact with a T cell, they polarize the microtubule organizing center towards the interface between the two cells, and this brings together with it the secretory apparatus very near to the interacting T cell. IL-12, which is a very important cytokine produced by DC upon an infection, is also brought very near to the T cell interface. If we interrupt this capacity to polarize by siRNA, by knocking down an important regulator of cell polarity, we've been able to prove that T cell priming is strongly compromised. But after this study, we became interested in studying a little bit more the identity of the vesicles that carry IL-12 in a cell. Because, I mean, as probably most of you know, IL-12 it's really important cytokine with pleiotropic function on the rest of the cells of the immune systems. And despite its important biological roles, there is very little known about how it's secreted out of the cell, how it travels, and what are the trafficking mechanisms to allow its secretion. So we inspired from work in macrophages, where a couple of cytokines trafficking pathways have been dissected, and we asked whether the cytokine is trafficked through endosomes of some kind, like lysosomes, late endosomes, or whatever. So to do that, to dissect out the trafficking pathways of cytokines, we focused on a class of proteins which are called SNARE, that are really specific and selective in allowing different transport steps between different organelles. So the first thing we did was to simply study by confocal microscopy where intracellular IL-12 is found to localize. And in here, what I'm showing you is in red, you see the 2 subunits of IL-12, which is a dimer, in cells that express MHC class II GFP. MHC class II is known to reside in endocytic compartments in dendritic cells. And we could find several structures in which the 2 proteins are highly colocalized. Probably most convincing, we use a dye to label the endocytic pathway, which is a dextran pH coupled to a pH-sensitive molecule that emits red bright fluorescence only when it reaches endocytic vesicles. And we could indeed find colocalization of IL-12, which this time is in green, together with the red signal of internalized dextran, proving quite strongly that indeed on its way out, dendritic cells intersect the endocytic pathways. We quantified, in order to identify which snare is implicated in the transport, we studied the localization between IL-12 and 3 different snares, which are prototypical of different trafficking steps: the trans-Golgi step, the early endosomes and the late endosomes, and we found that the strongest association is with the snare VAMP7, which is a snare that marks specifically late endocytic vesicles. So what does it happen to these vesicles when a DC encounter a T cell? In this movie, I'm showing you VAMP7 in red, and the T cell is in blue, that redistributes at the interface upon the interaction. This graph shows the peak in fluorescence at the synapse, and we can see that it clusters at the synapse immediately after a contact, then it goes away to go to a second T cell, and then it comes back to the first T cell. But to visualize directly the cytokine, we also expressed IL-12 tagged with GFP and took fast recording of vesicles moving in a DC that is interacting with a T cell, which is now indicated here with dotted lines. And I hope you'll be able to appreciate small dots of IL-12 that are moving very quickly here in the periphery, but there is especially a crowd of vesicles that is moving just underneath the synaptic membrane. And if we look at that in a high magnification, we can really see many vesicles that are docking and fusing with the synaptic membrane, altogether providing quite strong indication that indeed IL-12 is delivered at the synaptic, in the synaptic area. So what does that mean functionally? So in order to understand whether VAMP7 is really controlling secretion of IL-12, we used VAMP7 knockout, dendritic cells derived from VAMP7 knockout animals. We could show that indeed secretion of IL-12 is strongly decreased in DC deficient for VAMP7 expression, and especially and most importantly, when a DC which is full of intracellular IL-12 interacts with the T cell, the secretion that arises upon this contact, which is of this extent in wild-type cells, is totally abrogated in knockout cells. And when we measure T cell activation upon stimulation with wild-type or knockout cells, we also find a very strong impairment, indicating that VAMP7 is indeed the snare that controls trafficking of IL-12 for general secretion, like I'm showing in this summary, and during interaction with T cells. So I'd like to move now to our work in a totally different context, that is the immunodeficiency that I mentioned in the beginning. So we've got interesting in this syndrome because it is caused by mutation in a key regulator of actin polymerization. And given our previous findings on the role and importance of the cytoskeleton to favor the formation of the synapse, we realized that there was very little knowledge about the function of dendritic cells in this syndrome. So this is actually how we began to study what happens to dendritic cells when they miss this critical regulator of actin polymerization. There was a little bit of knowledge already at the time about the fact that lack of WASP obviously influences the capacity to internalize antigens and to migrate, but there was very little knowledge about the capacity of DCs to interact with T cells in this pathology. And I'm just summing up very, with just this slide, the experiments that we've made is to demonstrate that indeed DCs are affected in their capacity to migrate from the periphery to the lymph node in the WASP deficient, when cells are WASP deficient, and T cell priming is also highly impaired when it is performed by WASP deficient dendritic cells. Having these experiments and readouts well setted, we collaborated with a group in Milano who is developing and actually put in clinics the gene therapy for these kids. And we, in a preclinical model, show that indeed gene therapy treated animals rescue their capacity to migrate to the lymph node and to activate T cells. A second and quite different aspect of the disease that we studied is the role of plasmacytoid dendritic cells in the development of autoimmunity. This syndrome is basically an immunodeficiency, but kids also have a high frequency of autoimmune phenomena, and this aspect is the less understood aspect of the disease in terms of its pathology. I mentioned that disease exists in many subsets. One important subset are plasmacytoid disease, which are especially good in producing tons of type 1 interferon upon viral induction. So these type 1 interferons, which are obviously beneficial for a viral infection, can be detrimental in autoimmune diseases. And this has been proven in several so-called type 1 interferon diseases, like the lupus is probably the most well characterized. And because of some hints we had, we began to ask, we asked whether plasmacytoid disease in WASP may be involved in the development of the autoimmune phenomena. And I'm going to go quick on this part. We found that indeed in sera of the mouse model of the disease, we have an elevated level of circulating type 1 interferon at steady state, and when you isolate cells and perform real-time PCR, we do find spontaneous activation of type 1 interferon genes in the knockout, but not in the wild type. And when we isolate the cells and stimulate with various agonists of TLR, of TLRs, we again observe this tendency towards hyperreactivity in WASP-deficient cells. We've been able to confirm these observations in samples from human patients, where indeed we do observe hyperactivation of interferon genes, and when cells are isolated and stimulated with CpG, we observe the same hyperreactivity that we have first found in the mouse model. And finally, by crossing the mice to the WASP-deficient mice to IFNAR receptor deficient animals, so abrogating signaling towards the interferon receptor, we could rescue some of the autoimmune signs, overall proving that indeed in this syndrome autoimmunity is explained at least in part by a hyperactivation of plasmacytoid disease and the interferon pathway. We are now trying to understand why it is so, and we have some hypotheses given that WASP is an actin regulatory protein, and given that triggering of TLR9 occurs in endosomes, we think that all the effect we are observing has something to do with trafficking of TLR9 in the right compartment. And we are, yeah. And now I'd like to conclude with the last situation in which we studied dendritic cells. So we know that they are very good at presenting antigens and initiating immune responses, but it's also quite well known that during cancer progression, disease progressively lose their antigen-presenting capacities and actually turn into very bad cells that secrete immunosuppressive molecules that dampen the immune response. This is known, but the mechanisms are not really well understood. And this is why we really want to go and try to understand a little bit more. So far, we have just studied the model. Basically, we have some initial observations, but I'm just presenting to you the model just to know what we are up to. So we use, for now, animal models. We make transplant of lung cancer cells orthotopically in the lung of the mouse. We wait 14 days, then we harvest the tissues, and we have developed a very accurate strategy to identify different dendritic cell subset in lung tissues, and which is depicted here. But all I want you to notice is the CD103 and CD11b positive cells that I described to you in the beginning, which are typical subset of peripheral tissues. So what we do, we sort them out into two pure population, and we performed analysis on those cells by real-time PCR so far, because as you can imagine, these are really, really rare cells. So we end up with something like 3,000 cells, but still we can get some data out of it. So what Nicolette has done so far, which is a postdoc in my lab, is trying to follow what happens to this subset during cancer progression in terms of frequencies. And we observe that CD103 cells decreases, whereas CD11b cells increases. We don't know yet what that means. It's quite descriptive, but it's just, I mean, it has never been looked at before. In tumor draining lymph nodes, so she also harvesting the mediastinal lymph nodes and look at frequencies of the different subsets, CD103, CD8 alpha positive disease and CD11 positive disease, but there is no major change that we could evidence so far, but numbers are still, I mean, we have to go up with numbers. The thing which is interesting is that when these cells are re-stimulated ex vivo, we really observe a very important suppression of DC innate functions. So these are cells isolated from a control animal, from an healthy animal, and these are cells isolated from a lung of a diseased animals. These are cells known, well, there's one bar. Yeah, no, okay. So these are cells just taken out and non-stimulated. We are looking here at the levels of IL-12 produced upon stimulation with agonist of TLR9, TLR4, or TLR3. And as you see, cells from an healthy animals respond very well, but cells from tumor-bearing animals are really shut off in their capacity to respond to all three TLR agonists. And this is true for both CD103 and CD11 B cells, although the levels of cytokines produced by their subsets is relatively different. And we are now analyzing other genes that we hypothesize may be responsible for the loss of the capacity to respond to TLR engagement. And I think I'm done with this. I'd like to thank the past lab members that have done most of the work that I presented. Julian, Pulesio, Jelena, Francesca, Julia has just left actually, Asma and Nicoletta, which are still in the lab. And some of our collaborators and our founding agency. And I thank you for your attention.